My dear student friends, in today's lecture, let's study the integrated management of silkworm diseases. Successful silk production depends on the successful rearing of silkworms. One of the major constraints in silk cocoon production is the diseases in silkworm rearing. Mulberry silkworm, Bombyx mori, the primary producer of commercial silk, is domesticated for many centuries. The colonization has rendered the silkworms to lose its natural abilities to withstand the adverse climatic conditions and pathogen infection. The breed improvement programs aimed at evolving breeds with certain economic parameters have further eroded their ability to withstand pathogenic infections. This has made them vulnerable to many diseases, causing crop loss or low yield, which ultimately has led to low silk production. The microbes such as bacteria, virus, fungus and microsporidians cause infectious diseases in silkworms such as bacterial flagellum, nuclear polyhedrosis, cytoplasmic polyhedrosis, infectious flagellum and densonucleosis during all the seasons. Flagellum is caused by bacterial pathogens alone or in combination with infectious flagellum virus or densonucleosis virus. Grassery is caused by a viral pathogen BM-NPV. Muscardine and aspergillosis are fungal diseases which are common during winter and rainy seasons. Muscardine infects all stages of silkworms, whereas aspergillosis occurs during early instars of the larval stage. The occurrence of pebrine disease is rare and may break out at any period of the year. All these microbes have the potential to inflict significant loss in silk cocoon production. The cocoon loss due to diseases in India is estimated to be about 15 to 20 kg per unit of 100 disease free layings or 40,000 larvae. Though pathogens are the primary cause of all infectious diseases in silkworms, there are several inherent environmental and nutritional factors influencing survival of pathogen, the susceptibility of the host to infection and development of diseases. The inheritance of susceptibility to different diseases in silkworms is controlled mainly by polygenes and there is no breed completely resistant to all the diseases. For better understanding, the topic is being explained into different subheadings and they are: 1. Elimination of disease causing pathogens from the rearing environment. 2. Limiting disease spread in the rearing environment. 3. Use of disease free stock. 4. Preservation of contamination of mulberry leaf. 5. Timely disease diagnosis. 6. Disinfectants and bed disinfectants. 7. Rearing management and disease control. 8. Curative measures. Let us study these aspects one by one in detail. Elimination of disease causing pathogens from the rearing environment. The right approach to eliminate the pathogens from rearing environment is to conduct disinfection of the rearing environment prior to the commencement of silkworm rearing. Disinfection is an activity which results in the destruction of specific pathogenic microorganisms. Disinfection can be achieved by physical or chemical means or by both. Physical methods. The physical methods involve the use of sunlight, ultraviolet radiation, flame, heat and steams and boiling water. Sunlight. Sunlight acts as a disinfectant due to the heat it generates on the surface to be disinfected as well as due to the ultraviolet radiation emitted. The rearing equipments like stands, rearing trays, mountages etc. are disinfected by exposing them to hot sun. Attention must be paid to expose both the surfaces of the appliances. Many pathogens lose its infectivity when the contaminated rearing trays are exposed to hot sun for 20 to 30 hours. Ultraviolet radiation. Ultraviolet light are the electromagnetic radiation emitted by the sun which is having a wide range of wavelengths. Ultraviolet radiation of approximately 257 nanometers wavelength emitted by ultraviolet lamps is germicidal and can be used effectively to eliminate pathogenic microorganisms on the exposed surfaces and in air. The UV radiation disrupts DNA and RNA in living organisms. Flame or fire oxidizes the microorganisms into ashes. Though it is rapid, the threat of accidental fire and damage to the equipments and tools used for silkworm rearing is a limiting factor for the use of fire. Heat. It is an effective destroyer of pathogens. Dry heat destroys the microorganisms by oxidation and is non-corrosive and less effective than moist heat. Dry heat requires longer exposure time and high temperature. Moist heat or steam causes irreversible coagulation of microbial proteins. The action of moist heat is faster and more effective. Boiling water. Use of boiling water is an effective disinfection method. The maximum temperature obtainable is 100 degrees centigrade and the exposure of 10 to 30 minutes is effective, but this method is highly cumbersome and is not a reliable sporicidal. Chemical methods. Chemical means of disinfection adopts specific chemicals having the germicidal activity against targeted microbes for an assured and effective disinfection. The use of chemicals for disinfection gained momentum after Lister has showed in 1867 that phenol would kill microorganisms. The first criterion to be a good disinfectant is that the disinfectant should preferably have broad spectrum activity with the potential to kill microorganisms which are pathogenic to silkworms and spare non-pathogenic ones, as they help in the degradation of organic matter. An ideal disinfectant should be safe to use, non-irritating, free from toxicity and should not have unpleasant odor. Choice of chemical disinfectant depends on a number of factors. A. Number and nature of microbes to be destroyed. B. Type of surface of rearing house and appliances. C. Interaction with organic matter present in the surface to be disinfected. D. Contact time and temperature conditions. E. Toxicity to individuals, residual toxicity and effect on items such as fabric and material. F. pH, temperature, hardness of water being used, and finally cost. The second aspect of integrated disease management strategy is limiting disease spread in the rearing environment. For managing silkworm diseases in a silkworm colony, it is essential to follow hygiene practices. These are aimed at limiting disease spread in the silkworm colony and the prevention of secondary contamination and infection. It involves A. Personal hygiene, B. Rearing hygiene, and C. Application of bed disinfectants to prevent the spread of infection within the colony of silkworms. Personal hygiene. The personal hygiene involves the personal cleanliness of individuals involved in the rearing activities. Before and after entering the rearing room, hands should be thoroughly washed with a disinfectant solution and wiped with clean towel. The entry should be made into the rearing room by stepping on disinfectant solution or mat to avoid carrying pathogens into the rearing environment. Entry of unauthorized persons into the rearing house should be restricted for limiting the pathogenic movements in and out of the rearing room. Rearing hygiene. Rearing hygiene involves the orderly maintenance of rearing practices within the rearing room. Timely pickup and proper disposal of diseased larvae from the rearing bed is very important. Care should be taken while picking up of diseased larvae from the silkworm colony. It should be picked up with a chopstick or forceps and collected in a vessel containing disinfectant, and the same should be disposed appropriately. Care should be taken to avoid damage or physical injury to the silkworms while cleaning the bed. Similarly, during mounting of spinning larvae, Utmost care should be taken to avoid injuries to the larvae. C. Application of bed disinfectants. To avoid secondary contamination and spread of diseases in the colony, silkworm body and rearing seed disinfectants are to be applied. Periodic application of the bed disinfectants as per the recommended schedule significantly prevents the spread of diseases during rearing. Application of slacked lime powder at the beginning of each moult helps in absorbing moisture and regulates bed humidity. Slacked lime, being alkaline, is an effective virucide and highly helpful in managing the viral infections in the silkworm colony. Application of bed disinfectants after each molt is most effective disease management practice. General silkworm body and rearing seed disinfectants are antifungal, antibacterial, antiviral and antimicrosporidian in nature. Application of slacked lime dust in combination with bleaching powder in and around rearing house and premises improves hygiene in the rearing environment. Next aspect of our study is use of disease-free stock. Use of disease-free layings. Rearing healthy stock of silkworms ensures healthy crop. Silkworm rearing should be carried out using disease-free layings produced by authorized silkworm seed producers. The term disease-free layings indicates that the eggs are free from microsporidian or pebrine infection. Pebrine disease is unique in the way that it is transmitted from infected mother moth to the egg. To avoid transoviral transmission, mother moths after egg laying are microscopically examined for the presence of microsporidian spores, and in case they are found infected, the eggs laid by such moths are rejected. This process is called mother moth examination, which plays a crucial role in the management of pebrine disease. B. Surface sterilization. After accomplishing the mother moth examination and due certification procedures, the disease-free eggs are to be dipped in 2% formalin solution for 10 to 15 minutes. This primarily helps in exterminating the pathogens that are lodged on the surface of the eggs and ensures healthy larval colony. Another important aspect, that is the fourth one, is prevention of contamination of mulberry leaf. The quality mulberry leaf is vital for successful silkworm cocoon production. Therefore, the harvesting, transportation and storage should be done carefully to avoid contamination. Mulberry leaf is a good carrier of disease causing pathogens into the rearing environment. Over preservation of mulberry leaf should be avoided, as this could deteriorate the quality of feed, as well as there is a chance of higher level of bacterial multiplication in the leaf. Many mulberry pests are carriers of viral, fungal and microsporidian pathogens. The excreta and wing scales of these pests are a source of secondary contamination. Therefore, it is essential to control the mulberry and other agricultural pests in the mulberry garden. Timely disease diagnosis forms the fifth aspect of our study. During the course of silkworm rearing, it is essential to identify and diagnose the diseases at the earliest. This enables the removal of diseased larvae from the colony and curtails the spread of disease in the colony. In the early stages of infection, the signs and symptoms are atypical, and therefore the colony should be keenly observed, and any undersized or lethargic larvae, if observed, are to be picked up and disposed of properly. In the initial stages of infection, there may not be any visible external symptoms, and therefore microscopic examination of the larvae has to be carried out to find out the presence of any disease causing pathogen. Pebrine, nuclear polyhedrosis, bacterial flagellum and muscardine could be diagnosed and monitored following microscopic examination. Microscopic examination of homogenate of egg, moth, pupae, larvae and fecal matter for spore or its developmental stages is an appropriate approach to monitor microsporidian disease. For the identification of non-occluded viral diseases such as infectious flagellum and densonucleosis, specialized diagnostic approach is needed. As symptoms caused by these diseases are atypical at the early stage of infection and the pathogens are submicroscopic, immunodiagnostic method should be adopted. The sixth aspect of our study is disinfectants and bed disinfectants. Disinfectants for killing or inactivating pathogens causing disease in silkworms, various disinfectants are recommended. These are used to disinfect the rearing room, surroundings and rearing appliances apart from maintaining personal hygiene. Some of the disinfectants being used in silkworm rearing environment are formalin, sodium hypochlorite, quaternary ammonium compounds, iodine compounds, lime, phenolic compounds, calcium hydroxide, chlorinated lime, chlorine dioxide, organic chloric compound, sodium dichloroisocyanuric acid, trichloroisocyanuric acid, etc. The following are some major general disinfectants used in sericulture. Formalin. Formaldehyde in water is formalin and is known as formic aldehyde, methyl aldehyde, ethylene oxide, oxomethane and oxymethylene. It is a colorless gas and usually available as a 37% solution in water and methanol. It is clear and colorless solution with irritating odor, pungent and suffocating. Formaldehyde, the active ingredient in formalin solution is strong reducing agent. It forms formic acid utilizing the oxygen from the cells leading to the death of the germ. The efficiency of formaldehyde is directly related to the atmospheric humidity of over 70% and temperature of about 20 degrees centigrade. The gaseous nature of the disinfectant demands a closed condition for a longer period. Chlorinated lime, commonly known as bleaching powder, is a mixture of calcium hypochlorite, basic chloride, calcium hydroxide and freeze-lacquered lime. The active ingredient is hypochlorite. It is a white amorphous powder with a pungent smell of chlorine. For effective disinfection, an active chlorine content of 30% must be used. Hypochlorite in combination with weak acid and water produces hypochlorous acid and hydrochloric acid. Hypochlorous acid is unstable and resolves into hydrochloric acid and nascent oxygen. The oxidation reaction by nascent oxygen is germicidal. The hydrochloric acid also aids in the release of chlorine, which is also germicidal. Chlorine alters the membrane permeability of cells, allowing the micromolecules such as proteins and nucleic acids to leak out. The main component of chlorinated lime, calcium hydroxide, is germicidal, which dissolves the polyhedron protein to release and disintegrate virions. The action of bleaching powder becomes efficient under wet conditions and therefore the surface of walls and equipment should be drenched with this solution. Chlorine dioxide. Chlorine dioxide as gas is well known as most powerful antimicrobial agent and its advantage as a hard surface disinfectant is widely recognized. Chlorine dioxide is a yellow to green colored gas with a distinct odor similar to that of chlorine. Chlorine dioxide gas is highly toxic and very unstable. It has been stabilized into solution form, which is very effective disinfectant against pathogens causing diseases in silkworms. It is soluble in water and like oxygen it exists as gas in solution. Like ozone, chlorine dioxide reacts by chemical oxidation and do not form chlorinated organic byproducts. Stabilized chlorine dioxide is effective even at broad pH ranges. This stabilized chlorine dioxide is superior disinfectant than chlorine in many respects. It is stable, 2.5 times more effective than chlorine and 50 times than hypochlorites. It is a biotoxic agent causing the membrane damage. It reacts with the nucleotide and amino acid moieties of the germs. Calcium hydroxide, that is Silkworm lime. It is an antiviral agent and commonly used as a silkworm body and rearing seed disinfectant. Burned limestone calcium oxide is hydrated with water to give calcium hydroxide and Silkworm lime powder. The Silkworm lime, being alkaline, destroys the viruses and is most effective against BM-NPV, BM-IFV and BM-DNV. B. Bed disinfectants. It has always been a practice to use silkworm bed disinfectants to contain the spread of diseases during the progress of a silkworm crop. Several potential bed disinfectants have been developed in India for disease management during silkworm rearing. There are many popular bed disinfectants in the field for controlling various diseases of silkworms. To prevent secondary infection, occurrence and spread of diseases in silkworm colony, silkworm body and rearing seed disinfectants are dusted over the silkworm body and rearing seed following a schedule. Silkworm lime powder is dusted every time the larvae settles for molt. Other commercially available bed disinfectants are dusted while the larvae come out of molt and mulberry is fed after half an hour of dusting the bed disinfectants. The disinfectants are dusted at 3 to 5 gram per square feet bed area. It is important to note that the bed disinfectants do not cure silkworms from diseases and it only prevents the spread of diseases. Hence, it should be used as a preventive measure by dusting the powder formulation uniformly as per recommended quantity and schedule. The chosen bed disinfectant must be effective against all silkworm pathogens to prevent the spread of all diseases, that is grassery, flacheri, muscardine and pebrine. Certain precautions are essential while using bed disinfectant formulations. It should not be dusted on the silkworms settled for mold or under mold and on eatable mulberry leaves available in bed. While dusting, eyes, nose, ear and mouth are to be protected. Hands should be washed thoroughly after dusting. Now, let us see the seventh aspect of our study. Rearing management and disease control. Rearing platform. The adoption of suitable rearing method plays an important role in the prevention of infection and spread of diseases. Silkworms are best reared following platform rearing technique. The method is advantageous as it involves minimum handling of worms, leading to lesser chances of secondary contamination and retention of quality of mulberry for longer period. The feces and unhealthy and diseased larvae remain in the lower strata of the bed and hence further contamination process tends to be less. The platform rearing method offers three-dimensional spacing and good ventilation. Rearing environments. Managing the appropriate temperature, humidity and air current during the course of rearing is also very important for silkworm disease management. Ideal temperature and humidity conditions favor the healthy development of silkworms, which in turn resist the pathogen manifestation in silkworms. When the larvae become weak, it is easily susceptible to pathogens and hence providing ideal microclimatic conditions are important. High temperature and high humidity induced stress renders the larvae stop feeding and thereby become physiologically weak. This makes the opportunistic pathogens take upper hand due to weak resistance and the larvae become diseased. Similarly, buildup of high humidity in the rearing environment should be avoided as it facilitates the fungal pathogens to germinate on the surface of silkworm, penetrate the silkworm and finally cause the muscardine disease in the silkworm colony. Nutritious mulberry leaf. The mulberry of poor nutritive value will not be able to provide sufficient quantity of essential requirement to the larvae to produce antibacterial factor. This results in high rate of multiplication of infectious bacteria and development of Flachery disease. Weakness due to physiological starvation due to feeding of poor quality and insufficient mulberry leaf should be avoided. Therefore, it becomes imperative to provide nutritious mulberry leaf to reduce disease incidence. Now, the final aspect of our study, curative measures. There are several attempts to develop specific measures against different diseases such as chemotherapy and thermotherapy against bacterial, viral, fungal and microsporidian diseases. Amrut is an eco and user friendly botanical based formulation for controlling grassery and Flachery disease of silkworms. This is one of the effective curative formulation available to Indian sericulturists for saving their crops from diseases. The formulation can be mixed with water at 20 grams per liter and 70 ml of this solution should be sprayed to 1 kg of mulberry leaf and can be fed to silkworms. Thus, the topic can be summarized that the silkworm colonies under controlled conditions are susceptible to various diseases caused by bacteria, virus, fungus and microsporidians. By following different management strategies such as elimination of disease causing pathogens from rearing environments, limiting the entry of pathogens and prevention of contamination of mulberry leaf, the silkworm diseases could be prevented. Further, use of quality disinfectants and bed disinfectants, timely disease diagnosis, maintenance of rearing and personal hygiene, rearing management etc. help in integrated disease management in silkworm colonies. Thank you.